Maybe you've heard of the blood type diet, maybe you haven't. But in this video, we are talking about the blood type O diet.
For centuries, type O blood was dismissed as a simple clinical fact, a label on hospital charts with little significance beyond transfusions. But what if this unassuming trait carried hidden clues about ancient survival, long migrations, and the resilience of early human populations? Could a single blood type hold the key to understanding the forces, disease, environment, and adaptations that shaped communities across continents and millennia? Stay with us to know. The blood that wouldn't go away. For generations, type O blood was regarded as one of the simplest constants in medicine, a quiet, unassuming detail recorded on hospital charts, largely relevant only during transfusions.
But patients who have type O, they have no antigens on their surface, which is why type O can be the universal donor.
Its defining characteristic, the absence of A and B antigens, allowed it to bypass most immune defenses, earning it the title of the universal donor. In emergency rooms, this neutrality made type O indispensable. It could flow into nearly any recipient's bloodstream without triggering an immediate immune reaction, giving doctors a critical life-saving tool when every second counted. On the surface, this simplicity made type O easy to understand, and for decades, medical training treated it as such, a functional, predictable, and practical aspect of human biology. Yet the more researchers examined population genetics, the more this apparent simplicity began to crumble. When scientists expanded their focus from clinical use to the broader distribution of blood types across human populations, they noticed a striking phenomenon. Across the Americas, type O was not merely common, it was overwhelmingly dominant, unlike any pattern observed in other regions of the world. From the Arctic tundra of Alaska to the dense Amazonian rainforest, the prevalence of type O was remarkably uniform, a trend that extended across thousands of miles, diverse ecosystems, and isolated cultural groups. In certain indigenous communities, nearly every individual carried type O. Even groups that might have been expected to maintain genetic diversity through trade, migration, or intermarriage displayed astonishingly high proportions of a single blood type. Early explanations drew upon familiar concepts such as genetic drift or the founder effect, theorizing that if the first populations entering these regions already carried type O, its dominance could naturally have been amplified over generations. While these mechanisms could explain localized patterns, the sheer uniformity of type O across widely separated populations defied conventional reasoning. As data accumulated from thousands of samples, traditional explanations appeared increasingly insufficient. Genetic drift typically results in uneven outcomes, pockets of diversity, occasional anomalies, minor variations between communities. Across the Americas, however, the dominance of type O was so consistent, it seemed almost orchestrated. Communities separated by deserts, mountains, and rivers exhibited near-identical distributions. The likelihood that this pattern emerged purely by chance diminished with every new data set. Some researchers began to propose a more provocative, though unverified idea, that ancient selective pressures may have preserved type O while gradually diminishing A and B antigens across entire populations. Although no specific cause has been confirmed, the consistency of type O suggested that natural factors, rather than randomness alone, had shaped its prevalence. These could have included environmental pressures, disease exposures, or other survival challenges faced by early populations. Archaeological evidence strengthened this hypothesis. Ancient DNA, extracted from skeletal remains across the Americas, revealed that type O dominance was not a recent development. It extended far back in time, predating European colonization and persisting across centuries of changing climate, migration, and population shifts. This indicated that type O had been deeply embedded in the genetic structure of early human populations from the Americas. Mapping these ancient distributions highlighted a subtle but profound truth. The story of type O blood is far more than a clinical or physiological observation. It represents a genetic echo of deep history, a relic of the forces that shaped the earliest populations of the Americas. These patterns hint at events of remarkable complexity, involving migrations, selective pressures, and adaptive survival strategies, all leaving traces in a blood type once dismissed as simple. Type O, once treated as merely functional, emerges as a marker of human resilience, persistence, and the enduring legacy of ancestral populations navigating challenges across millennia. What force, natural or environmental, could have shaped early populations so dramatically that only the resilient type O bloodline endured and thrived across a number of generations? Cracks in the ice bridge theory. For decades, the story of the first Americans was presented as one of the cleanest, most straightforward narratives in anthropology. According to this long-standing model, humanity entered the Americas roughly 15,000 to 20,000 years ago by crossing Beringia, a vast land bridge that once connected Siberia to Alaska during the last ice age. Textbooks and popular accounts depicted small bands of hunter-gatherers following megafauna herds across a frozen corridor, eventually dispersing throughout the continents as rising seas submerged the land bridge behind them. This framework painted a picture of orderly migration, suggesting that the settlement of the Americas was a relatively simple event, defined by a single corridor and a limited founding population. Within this model, it was assumed that these early migrants represented only a small fragment of Siberia's genetic diversity, carrying with them whatever traits happened to exist in their small founding group. If, by chance, this population included mostly type O carriers, then the dominance of type O in modern indigenous populations of the Americas could be explained as a natural consequence of founder effects. On the surface, the theory seemed coherent. A tiny group carrying mostly type O crosses into a new continent, reproduces over millennia, and spreads their blood type widely across North and South America. However, access to ancient DNA began to challenge this tidy explanation. Geneticists analyzing remains from prehistoric Siberian populations discovered that these groups were far more diverse than previously assumed. Their genetic profiles included all three major blood type alleles, A, B, and O. Far from being uniform or narrowly limited, these populations exhibited diversity comparable to the broad spectrum observed in modern Eurasia. This finding directly contradicted the idea that the earliest migrants carried almost exclusively type O. If the first Americans truly inherited this wide genetic diversity, then A and B alleles should still be detectable in both ancient and modern populations across the continents. Yet extensive genetic sampling revealed a puzzling reality. Across vast regions of the Americas, A and B were nearly absent, sometimes undetectable. The uniformity of type O was far too pronounced to be explained solely by genetic drift, which typically produces uneven, scattered patterns rather than continental-scale uniformity. Faced with this discrepancy, scientists began to reconsider the simplistic founder population explanation. The overwhelming absence of A and B alleles suggested that early populations in the Americas may have experienced a genetic bottleneck, a rapid reduction in diversity triggered by a powerful selective force. While the specifics remain unverified, some researchers speculate that environmental pressures, disease, or other biological factors could have disproportionately affected carriers of A and B, leaving type O as the surviving lineage. Others suggest that cultural or dietary factors may have played a role, though no concrete evidence confirms any particular mechanism. These findings reveal that the classic ice bridge narrative is far too simplistic. Early American populations did not merely migrate and settle in isolation. They underwent dramatic genetic changes after arriving in the Americas. What forces could have driven such sweeping transformations? And how did these shifts shape the survival of entire populations? If ancient migrants carried all blood types, what dramatic event or pressure eliminated nearly everything but type O across vast territories? The vanishing bloodlines. By 2025, a coordinated international effort among researchers in Canada, Brazil, and South Korea had launched one of the most ambitious ancient DNA mapping projects ever undertaken in the Americas. The objective was clear: to trace the presence and distribution of type A, type B, and type O alleles across thousands of years of human history. Yet, what the team discovered went far beyond expectations, revealing a pattern of blood type evolution that challenged long-held assumptions about how these alleles spread or disappeared throughout the hemisphere. The earliest skeletal remains analyzed came from Alaska, geographically closest to the original entry point from Siberia. Genetic testing revealed faint but undeniable traces of type A, demonstrating that the initial populations entering the Americas were not exclusively type O, as some earlier theories had suggested. This finding confirmed that multiple blood types had crossed the Beringian land bridge, establishing a baseline of genetic diversity in the continent's earliest settlers. However, as researchers extended their survey southward, the genetic landscape began to shift dramatically. In samples from the central plains of North America, traces of type A disappeared entirely. Further south, in regions corresponding to Mesoamerica, ancient remains revealed the same striking uniformity. By the time scientists examined early skeletal material from the Andes and farther into South America, both type A and type B had vanished entirely, leaving type O as the only detectable allele in these populations. This continent-wide disappearance was unlike the gradual, uneven changes typical of genetic drift, which usually leaves behind scattered pockets of diversity. Instead, the data suggested a rapid and sweeping reduction in blood type variation, almost as if entire lineages carrying A or B alleles had been eliminated within a relatively short span of time. The speed and scale of this pattern prompted researchers to consider mechanisms beyond ordinary population dynamics. Several hypotheses emerged, ranging from established biological processes to unverified, alleged, or even rumored causes. One possibility centered on an ancient pathogen that may have exploited A or B antigens as entry points, conferring a survival advantage to type O individuals during widespread outbreaks. Another hypothesis proposed unknown environmental pressures, possibly related to diet, climate, or immune response.
A blood type O person do better with chicken and beef and a list of other high protein food versus carbohydrates or fat.
That indirectly disadvantaged carriers of A or B alleles. Others speculated about extreme survival events, such as prolonged famine, catastrophic climatic shifts, or other stressors that may have selected for immune profiles consistent with type O. A pivotal study summarizing these patterns described the event as a directional selection, implying that the loss of blood type diversity followed a specific trajectory rather than occurring randomly. While the precise trigger for this selective push remains unverified, the outcome was unmistakable. Populations once carrying multiple alleles became overwhelmingly dominated by type O, a genetic signature that persisted across continents and millennia. The long-term effects of this transformation are evident even today. Type O evolved from a mere clinical descriptor into a living testament to the survival of populations that endured trials whose details have largely been lost to history. It became a marker of resilience, a genetic echo of ancient populations navigating environmental, biological, and social challenges that remain largely mysterious. Even as modern researchers continue to debate the precise causes, the pattern is clear. A dramatic, near-total narrowing of genetic diversity left type O as the surviving allele in many regions of the Americas. This evidence highlights the complexity of human adaptation. Rather than a simple story of migration or chance, the prevalence of type O reflects selective forces acting across vast populations, shaping the genetic landscape in ways that textbooks had never anticipated. Whether driven by pathogens, environmental pressures, or catastrophic events, the result is undeniable. Type O stands as the enduring signature of the America's earliest inhabitants, a reminder that the past contains mysteries far deeper than even the most extensive archaeological surveys can fully explain. If type O carriers endured a survival trial, what biological advantage helped them persist while other bloodline markers vanished from the continent? The immunity rift. As researchers delved deeper into the peculiar dominance of type O blood, their focus shifted from migration patterns and ancient DNA toward a more intricate area: immunity. Modern medical and epidemiological data, gathered from diverse regions across the globe, revealed a subtle but persistent trend. Individuals with type O blood often exhibited slightly reduced severity in several major infectious diseases, including cholera, malaria, dengue fever, and certain severe respiratory infections. While this advantage was modest, it was measurable across large populations. Type O carriers were not invincible. They could still suffer grave illness or even death. Yet, when researchers compared survival outcomes over decades and centuries, the data suggested that type O individuals fared marginally better than average in the face of widespread disease. Even a small survival advantage on an individual level can have profound consequences over the course of hundreds or thousands of years. Before the advent of modern medicine, vaccines, or reliable sanitation, even a few percentage points of increased survival could gradually shift the genetic composition of entire communities. The effect is amplified in regions with harsh environmental conditions or frequent exposure to infectious outbreaks, where resilience can determine which populations endure and which decline. Nowhere was this long-term influence more apparent than in the Andes, one of the most extreme and unpredictable human habitats on Earth. High-altitude Andean populations have endured millennia of environmental stress: thin air, fluctuating temperatures, intense ultraviolet radiation, and persistent exposure to pathogens. Genetic studies in these communities uncovered a strikingly high prevalence of an immune-related variant known as IL-1752C. This gene variant plays a crucial role in regulating inflammatory responses, helping the body manage the fine balance between fighting infection and avoiding uncontrolled immune reactions, such as cytokine storms, which can be rapidly fatal in severe disease. What made this finding particularly noteworthy was the frequent co-occurrence of type O blood and IL-1752C within Andean populations. The association appeared far more often than would be expected by chance alone, suggesting a potential biological link between blood type and immune modulation. Researchers proposed a tantalizing but unverified hypothesis. Ancient populations may have repeatedly faced survival pressures, whether from infectious disease, environmental extremes, or unknown factors that favored individuals carrying both traits. This combination could have conferred a subtle but meaningful advantage, improving survival during times of crisis. Although these connections remain speculative, they offer a window into a deeper narrative of human adaptation. Type O blood may not simply represent a blood group, it could be part of a broader resilience profile shaped over millennia. Its frequent pairing with IL-1752C hints at a genetic signature left by sustained exposure to extreme conditions and repeated selective pressures. In this view, the co-occurrence of these traits becomes a biological echo of ancient survival challenges, encoded within the genomes of populations whose ancestors endured environments harsh enough to influence evolution permanently. The implications of this research extend beyond the Andes. It suggests that blood type, long considered a neutral clinical feature outside of transfusions, may have played an active role in shaping population-level survival. If future studies confirm these associations, type O could be seen as more than a universal donor. It might also reflect the enduring imprint of ancient selection pressures, preserving subtle yet vital advantages across generations. The presence of IL-1752C alongside type O remains a powerful reminder that human evolution is not only recorded in skeletal remains or migration patterns, but also in the quiet, persistent signatures embedded in our genomes. Viewed through this lens, type O blood and associated immune traits become living testaments to resilience, adaptation, and the hidden biological strategies that allowed early populations to survive environmental and infectious challenges that modern humans can barely imagine. If type O carries hints of ancient immune battles, what deeper genetic memories might still lie hidden in humanity's oldest bloodlines? The Lake Bowl Marker. In the remote, frozen expanse near Lake Bowl in Siberia, archaeologists unearthed a remarkably well-preserved human skull, estimated to be more than 12,000 years old. The permafrost acted as a natural vault, maintaining the integrity of fragile tissues and DNA fragments that modern sequencing technologies could finally analyze. Scientists anticipated that examining this ancient genome would enrich our understanding of early northern populations, particularly those inhabiting the harsh landscapes of Ice Age Siberia. Yet, what they discovered went far beyond expectations and deepened the ongoing puzzle surrounding type O blood. Within the ABO gene cluster, geneticists identified a peculiar feature described as a rare deletion mutation, labeled O delta variant 1. Unlike the typical single nucleotide changes responsible for most blood type variation, this mutation involved a missing segment of DNA, a more substantial alteration that often emerges under intense evolutionary pressures. If the characterization is accurate, such a deletion would not be a random anomaly. Instead, it could signify that the individual belonged to a lineage exposed to extreme biological or environmental stressors, possibly selecting for traits that would later influence blood type prevalence. This variant was allegedly absent in ancient remains from Europe, Africa, and most East Asia, making its presence seemingly unique to this Siberian specimen and certain indigenous North American groups. This proposed exclusivity immediately drew attention because it suggested a potential genetic bridge between Ice Age Siberia and later populations in the Americas. The survival of a mutation across thousands of years and vast distances could indicate a remarkable continuity of ancestry, hinting at ancient migration patterns that connected continents in ways previously unconfirmed by the archaeological record. However, the broader scientific community has yet to verify these claims. Other reports suggesting that this deletion contributed to the rise of type O dominance remain unconfirmed. No experimental evidence has demonstrated a functional connection between the mutation and blood type expression, immune resilience, or disease resistance. Until independent research corroborates it, the significance of this finding must be considered speculative. Even as a hypothetical concept, the suggested connection is striking. If the Ice Age Siberian individual did carry a deletion that later emerged in certain North American groups, it would imply a remarkably durable genetic thread, one that survived waves of migration, shifting climates, and millennia of survival challenges. This mutation, if real, functions metaphorically as an ancestral fingerprint, marking a lineage that endured while other genetic markers faded or disappeared. Even without verification, the narrative resonates with the broader mystery of type O blood dominance. The presence of such a deletion could provide a speculative mechanism for why type O persisted when other alleles vanished. Early carriers may have experienced catastrophic pressures, pathogens, environmental extremes, or dietary challenges that selectively favored individuals with this deletion. Over generations, such selective forces could have contributed to the sweeping predominance of type O observed in many indigenous American populations. While the role of O delta variant 1 remains uncertain, the scenario it presents offers a compelling story of human survival and adaptation. It suggests a lineage shaped by adversity, a bloodline that endured ice age conditions, crossed continents, and retained its genetic signatures across epochs. Whether or not future studies validate the existence or function of this deletion, the concept underscores the profound complexity of human genetics, highlighting how subtle genomic features may reflect histories far older than written records.
They looked into the human genome and they found what appeared to be an extraterrestrial stamp on our genetic code. And it operated very much like a mathematical type of code.
And hinting at the enduring legacy of type O blood as a marker of resilience and continuity. If an ancient Siberian genetic imprint survived migration and catastrophe, what other buried clues might reshape our understanding of early human survival? The Crawford revelation. In early 2025, a routine contribution to an ancestry mapping project unexpectedly opened one of the most controversial chapters in the ongoing investigation of type O blood. Thomas Crawford, a member of the Blackfeet Nation, volunteered a DNA sample with the simple goal of learning more about his lineage. The project itself was unremarkable, one of many community-driven initiatives intended to document genetic heritage across indigenous populations. What made Crawford's sample extraordinary, however, was what researchers reportedly discovered within it. Crawford's genome contained the same O delta variant, one deletion identified in ancient remains near Lake Bol in Siberia. If accurate, this finding would suggest a direct ancestral link spanning more than 12,000 years of human history, connecting an Ice Age Siberian lineage with a contemporary indigenous American individual. While this connection remains entirely unverified, the mere possibility generated immediate excitement among the research team. The idea that a mutation described as both ancient and rare could appear in a modern genome was unexpected, raising questions about the persistence of certain genetic traits over millennia. Yet the most provocative discovery came from a different portion of Crawford's DNA, an area near the HLA region known for its role in regulating immune responses. Researchers detected a small sequence previously classified as junk DNA.
But those genes only make up about 1 to 2% of our DNA. The other 99% of our genome is non-coding, since the term junk DNA was coined in the 1960s.
A term used for genetic fragments with no established function. For decades, this fragment was considered insignificant. However, when Crawford's data were processed through advanced simulations of immune response, the sequence displayed behavior that no one anticipated. Under extreme biological stress, conditions simulating severe infection, multi-organ dysfunction, or oxygen deprivation, the fragment allegedly activated in a sudden, coordinated manner. Unlike a weak or partial response, it reportedly triggered a cascade capable of regulating inflammatory pathways, potentially preventing catastrophic immune collapse. The scientists chose to refer to this as an emergency survival switch, though its function remains unverified. Peer-reviewed research is yet to confirm such a mechanism, and the researchers themselves emphasized that the role of this sequence, if any, is unknown as of yet. Adding to the intrigue was a whistleblower claim that added more pieces to this already expanding puzzle. According to an anonymous source, a similar fragment had been flagged during early analyses of the Human Genome Project in the late 1990s, but was allegedly removed from public datasets. This assertion, though lacking in documentation, has managed to fuel speculation and raised questions about whether unusual genetic features may have been overlooked or deliberately omitted in earlier research. Perhaps the most compelling aspect of Crawford's sample was its reported association with type O blood in certain indigenous populations. Although alleged, the idea suggested a potential convergence between ancient genetic signatures, unusual immune features, and blood type patterns that appear to have persisted across thousands of years. If real, this convergence could indicate that type O blood was not simply a matter of chance or founder effects, but may be linked to broader survival mechanisms embedded in human genomes. Whether coincidence or evidence of a deeper biological narrative, Crawford's genome became a focal point in the ongoing effort to understand why type O persisted while other alleles disappeared. The combination of the O-delta variant 1 deletion, the unusual HLA-associated fragment, and the alleged immune-modulating properties created a complex puzzle. Researchers now faced the challenge of disentangling unverified claims from reproducible data, while exploring the possibility that ancient genetic pressures, environmental stressors, and immune adaptations may have intersected to favor type O over millennia. If a rumored defensive switch lies hidden in certain genomes, what global connections might it reveal across ancient populations separated by oceans? The Polynesian Passage. The genetic landscape of the Pacific Islands, particularly Rapa Nui or Easter Island, offers intriguing insights into the broader story of human migration and adaptation. In 2025, excavations on a remote island uncovered skeletal remains dating back roughly 900 years. These bones, remarkably preserved in the volcanic soil, were sent to genetic laboratories for detailed analysis. The findings surprised researchers. Not only did the remains display an overwhelming prevalence of type O blood, but they also carried the ILD-17 variant 52C, an immune-modulating gene variant previously identified in highland Andean populations thousands of miles away in South America. The coincidence of the same rare immune variant appearing in both locations was striking. Researchers noted that the odds of this happening purely by chance were extremely low, suggesting the possibility of direct genetic exchange. This, in turn, implied that Polynesian voyagers may have interacted with South American populations centuries before European contact, though this idea remains debated and not universally confirmed. For years, the notion of pre-Columbian contact across the Pacific had been discussed in academic circles, often based on similarities in crops, artifacts, and oral traditions. The DNA evidence seemed to provide a potential biological counterpart to these cultural indicators. If Polynesian navigators reached South America, they may not only have transported goods and ideas, but could also have carried genetic variants that influenced immunity, including traits that enhanced survival against infectious diseases. While the findings suggest intriguing possibilities, the interpretation of DNA as evidence of direct contact is still speculative and unverified. Meanwhile, another area of research was unfolding in the Andean highlands. Archaeologists examining dental enamel from ancient individuals identified tiny traces of antibodies, microscopic records of immune responses. Some of these antibodies appeared tuned to pathogens that are no longer present in modern populations, likely belonging to extinct diseases long erased from human experience. Most of the individuals displaying these immune markers carried type O blood, while other blood types were rarely observed in these samples. The implication was that type O may have conferred a survival advantage during outbreaks or environmental stress, though this interpretation is based on observed correlations and remains unverified. The pattern suggested a grim reality. In periods of severe epidemic or extreme environmental challenges, type O blood may not have been just common. It could have been the blood type most likely to endure. Whether through a combination of natural immunity, genetic resilience, or environmental adaptation, type O repeatedly outlasted other blood types, becoming more entrenched in the surviving gene pool over successive generations. Though further research is required to confirm these claims, the apparent Polynesian connection introduced a further layer of complexity. If correct, it would mean that the spread of type O and its associated immune traits was not limited to land migrations across glacial corridors, but could also have been transmitted across vast oceanic distances. Voyages may have been motivated by exploration, trade, or alliance, yet they also potentially acted as a conduit for genetic traits that had historically supported population survival. While DNA analysis suggests this possibility, direct evidence confirming such voyages or genetic transfer is not fully established. This emerging picture, while still mostly unconfirmed and alleged, extends the narrative of type O blood beyond clinical relevance. From the Amazonian jungles to the volcanic soils of Easter Island, and from the Andean highlands to Siberian permafrost, the genetic markers associated with type O appear repeatedly. The co-occurrence of IL-1752C in geographically distant populations raises questions about ancient selective pressures, immune resilience, and patterns of human survival that remain largely unexplained. What emerges is a complex story in which type O blood intersects with immune traits, geographic migration, and potential cross-cultural contact. The skeletons from the island provide evidence of both genetic prevalence and immune adaptation. They suggest patterns and possibilities, but many of these remain open questions. The repeated presence of type O blood and the IL-17 variant 52C in distant populations underscores the potential interplay of environment, disease, and human mobility. Yet, the exact historical and biological mechanisms remain unresolved. Each discovery hints at a broader narrative of endurance, selection, and genetic continuity, while leaving significant uncertainty about the causes, timing, and nature of the connections across the Pacific and South America. Type O blood emerges as more than a trait. Could it be the lingering mark of ancient trials, a testament to humanity's resilience across time, space, and unimaginable challenges? Share your thoughts in the comments below.